what WHO has shown recently is that implementation of new laboratory techniques has led to exponential increase in the number of cases diagnosed of multi-drug resistant tuberculosis, and I guess I'll try to explain what technologies have been used to increase the rate of MDR-TBs and how they can inform management of patients. I would like to start with another WHO slide that captures estimates of MDR incidence across the globe. The size of a bubble represents the number of cases, and you can see the largest bubbles are obviously in our region, especially in the subcontinent and in China. So India has the largest number of cases, and we all understand the reason for that. Then if we look at New South Wales TB control program documents, and Ellen has put together this report from New South Wales data between 2011 and 2014, capturing the country of origin of TB cases, and the majority of TB cases are diagnosed in patients born overseas in high incidence country. Again, in India stands out as a leading contributor to this problem. Not surprisingly, we've started making connections with our colleagues in TB research in India, and Ben, Marais, and Shailendra. I've just returned from a trip to India, and this is a picture of participants of the workshop on tuberculosis and genomics, which was hosted by our partners in National Institute for Research in Tuberculosis in Chennai. You can recognize Ben is here in the shade, the best place. I think it was plus 40 at the time. We barely survived this 3 minutes of photo outside. This is Shailendra, director of the institute, and many other participants that represented all major microbiome reference laboratories in India. And I would like to thank Ben and Shailendra and Daniela Somers, who is the director for Global Health Office at the Faculty of Sydney Medical School, for making this trip possible. You might remember and recognize Nathan Beckman, who is a CRE-TB postdoc. And he and Elena Martinez, who was a CRE-TB postdoc, not that distant past, doing hands-on training in microbiome tuberculosis genomics analysis with representatives from these laboratories. You might know that Prime Minister of India made an announcement and huge commitment to eliminate tuberculosis in India by 2025. This elimination means that moving from 2.5–3 million cases per year, and roughly, of disease diagnosed every year to 200,000 per year. So it's not a maybe elimination in a context of Australia. Nevertheless, as a result of this big initiative, a lot of resources have been contributed to the improvements in diagnosis of TB, especially of multi-drug resistant tuberculosis, and capacity in genomics has been recognized as a core one. And MySeq machines have been procured to laboratories in Delhi, Bangalore, Mumbai, Chennai, and now bioinformatics analysis of genomes has become a major bottleneck, and this is a gap that TBCRE, and we as a group here in Sydney are trying to assist our colleagues in India with. Genomics represent one extreme, one end of MDR detection. The more simple and rapid methods of MDR detection are PCR based, and a lot of activity has started with the endorsement by the WHO of GeneXpert, and more recently Expert Ultra platform with a bit better sensitivity and specificity. And in addition to this PCR black box, which is a GeneXpert, several other PCR assays automated to lesser or more extent have been introduced already and available in the market, produced by different companies in different countries. GeneChip is one example by Capital Bio in China, MELPro, GeneDrive by Epistem, and more recently an Abbott assay. And in addition, I haven't listed Korean assay and a new assay from Bangalore produced in India. So essentially, success invites imitation, and the hope is that these multiple platforms will eventually drive the cost down, and they have fairly similar performance characteristics, or they're likely to have very similar performance characteristics. Even before GeneXpert, line probe assays again have been endorsed by WHO as a method of MDR TB screening. Started with the Hain assay or INOLIPA assay initially, and now with other assays that are based on PCR detection in the first step, and then reverse line blood hybridization in a second step, where resistance to isoniazid and rifampicin and some other drugs can be detected from cultures as well as directly from clinical specimens. And this slide shows cumulative performance characteristics, forest plots from meta-analyses that combine sensitivity, that analyze sensitivity and specificity of line probe assays, and the approaching sensitivity and specificity of GeneXpert. I think sensitivity of 90-95% is probably based on just smear positive samples in a high incidence country. This end of spectrum, which you can see my pointer, in low incidence countries, and specificity is very, very high. I wanted to show this one table from a recent study done by researchers, which was, I'm sure, partially funded by Abbott, but it's a nice comparison of Hain assay as a one probe assay, GeneXpert platform, and another PCR-based assay, in this case Abbott one, on a relatively small number of samples from Bangladesh, tested in a lab in Germany. But this slide highlights very, very similar characteristics as far as sensitivity and specificity of those kits on clinical samples, and I think quite realistic. So this is sensitivity 98% and specificity around 87%. It's very hard to move any further in terms of detection of TB and MDR-TB using PCR-based assays. Specificity has been recognized as a big issue in PCR-based assays for patients partially treated with tuberculosis, and it has been highlighted with ultra assay. as well, and recently this report from Africa has pointed out the importance of testing mutations outside the conventional sets of target genes, where gene experts missed essentially up to 30% of TB or rifampicin-resistant isolates due to mutation in RPB gene sitting outside the conventional rifampicin resistance defining region. So this is a false negative MDR cases due to design of gene expert. So drug resistance detection has been redefined because of these considerations and concerns, and acknowledgement that we certainly learned a lot about molecular markers of drug resistance that are placed in different genes and across the Mycobacterium tuberculosis genome, and genome-wide analysis has more power in describing and capturing the drug resistance than PCR-based, PCR-focused assays. And I probably shouldn't be spending too much time on describing each individual gene target that is targeted by first and second line drugs. And I'll use an example of RPAB gene and pyrazinamide resistance to illustrate the point. So this is rifampicin resistance defining region in RPAB gene in Mycobacterium tuberculosis, just 81 bases, and each codon, 3 bases, can be labeled by using TB system or E. coli genome system, and unfortunately it creates sometimes a bit of confusion. But nevertheless, you can see that majority of mutations leading to phenotypic resistance to rifampicin are confined to this cluster 1 region, 512, 534 codons in E. coli classification. Since the description of this region is mutations in cluster 2 regions, 563, 574, and cluster 3 codons 687 have been recognized as important contributors to rifampicin resistance, but clearly they would be missed by a conventional expert system. These mutations lead to change in the binding between rifampicin molecule and the target in RPAB, and as a result of this, translation and transcription would be affected, and bacteria would survive the effect of the drug because of conformational changes in the protein. This in each codon there could be different types of mutations. We have pretty good clues now in our catalogs what kind of mutations should be expected and are present in different mycobacteria. So our accuracy of detection is quite high, and it has been recognized that spectrum and frequency of distributions of these mutations is very much dependent on the lineage, and as a result of this, would vary significantly between different locations in the world. And you can see that in South Africa, mutations in rifampicin resistance defining region will be present in 71% of cases, and in Thailand in 100% of cases. But there is a big variability, and this is just a subset of studies, obviously with a biased sampling strategy. The story of pyrazinamide resistance is quite quite different because PNCA gene is a major target of pyrazinamide resistance, and you remember that pyrazinamide is a prodrug, and it's kind of transformed into active compound by enzyme which PNCA gene codes for pyrazinamidase, and this pyrazinamidase production can be reduced by mutations in different parts of this gene. There is no particular hotspot. These mutations can occur in different parts of the gene. The gene is quite large in size. The gene can be excised completely on its own, or part of the whole 10, 15, 20 kilobases of genomic material can be left out, and this would lead to resistance to pyrazinamide in isolates from Korea, from China. This is what we've seen in our lab in Sydney. And if you look at the distribution of frequencies of PNCA mutations, again, these frequencies are much lower than RPOB mutations for rifampicin resistance defining region. So there is a bigger role of mutations in other genes, which we're just starting to learn about. Again, re-emphasizing the point about genome-wide analysis for mutations. I might have to move without my glasses to here, to stand in front of you and just illustrate the point about relationship between different mutations and the actual MIC phenotypically. So this is this analysis of a small subset, 25 MDR-TB isolates, using examples of rifampicin and INH. Cutoff rifampicin susceptibility is around 1 milligram per liter. And you can see that isolates with no mutations, wild type or reference strain, would have much lower MICs, and isolates with mutations, yellow and blue, and this is RPB codon mutations 545, 534, would have much higher MIC. And this purple is example of mutations outside of rifampicin resistant defining region. So this is clearly type of strains that would present significant problem for laboratory using current methods to screen for MDR-TB phenotypically. So this is a not trivial task to identify low borderline resistance, which would lead to higher risks of clinical failure on first-line regimen. The same applies to INH, isoniazid. Again, strains with no mutations clearly show very low MIC phenotypically, and strains with mutations in CAT-G and INH A genes would have elevated MICs in this example with a high level resistance to isoniazid and cross-resistance to some other drugs. And this relationships between clinical outcomes and mutations are now used to design clinical trials, and clinical trials are underway looking for a relationship between CAT-G, classical mutations, high confidence mutations, and the failure and success on alternative regimens. And because our catalogs are becoming more extensive, we can certainly grade confidence in detection of drug resistance, and mutations that are associated with significant increase in MIC with a likelihood ratio of greater than 10 are defined as high confidence mutations. And so we know these mutations in CAT-G gene and INHA. We have a list of high confidence mutations in our POB gene, as well as mutations with a lower confidence of association between genomic and phenotypic data. for PNCA gene, for ethambutol. Interesting thing is that changes in a same codon in RRS gene lead to cross-resistance to amikacin and capreomycin, for example. And 8 codons downstream, it will be just mutation leading to amikacin resistance only. 1402, sorry, 2 codons downstream. Just capreomycin resistance, no amikacin resistance. So granularity of our understanding and predictive power has increased in the last 5 years dramatically, as well as appreciation that some changes in some mutations could code for cross-resistance between isoniazid, for example, and ethionamide and prothionamide. I'm referring to NH gene. So, major international studies, and there are at least 3 now published, and Cryptic is the largest consortium of 25 countries, and majority of these countries are high incidence countries with tuberculosis, collecting and analyzing data on thousands of isolates fully sequenced. Majority of them are susceptible isolates, but there are at least more than 2,000 resistant isolates with very high accuracy of prediction of resistance, 96.5% sensitivity and 98.4% specificity. Adding machine learning algorithms to infer resistance added extra 1.52% to sensitivity parameters. So we're clearly in a range of clinically actionable resistance prediction and inferences from data provided by whole genome sequencing. And I'm nearly done, but I would like to use the example of bedequillin resistance to illustrate some final points. In this study that has been done recently by Elena Martinez, as I mentioned, she's been working with support from CRE-TB, and because we've been sequencing mycobacterium tuberculosis in our reference laboratory at ICPM Out Westmead since October 2016, we've been in a position that allows us to look retrospectively at markers of resistance to second-line drugs. We haven't seen bedequillin resistance phenotypically prior to this study, but what we wanted to do, we wanted to check markers of bedequillin resistance in MDR cases first, in all MDR cases that we've sequenced over the last 10 years. And we found mutations in ATP-E gene, which is associated with high-level resistance to bedequillin, as well as mutations in RV0678 gene or protein coding region, which had been associated with low-level resistance to bedequillin, often with a cross-resistance to clofazimine. So what was interesting to see the relationship between phylogeny, between the phylogenetic properties of strains analyzed and distribution of these mutations. So on this graph, you can see that majority of on the outer ring, Beijing lineage strains are in yellow, and East African Indian strains, predominantly from the subcontinent, are in red. So you can see ITP genes, high-level resistance defining mutations are primarily in Beijing isolates and low-level resistance to bedequillin in East African Indian and European isolates. And majority of these strains with mutations, when we did phenotypic susceptibility for the vehicle, appear to be phenotypically susceptible, but we did see several isolates with elevated MIC to bedequiline. So phylogeny of mycobacteria, origin of this mycobacteria, would have significant implications for interpretation of drug resistance. So I think the strategies that CRE TB has been chosen for many years, focusing on countries where TB originate, where TB cases are coming to Australia, helping them with the capacity building, doing projects together. I think it's a very wise strategy and should deliver a lot of public health benefits to Australia. We're destined to see more and more MDR-TB because more and more testing is done, both by PCR-based methods and by whole genome sequencing. In India, where they've implemented GeneXpert now statewide, and so where they're rolling out now whole genome sequencing, rates of MDR-TB are just astounding, 30-40%. So we will see more and more of this very difficult to treat cases, and from data management point of view, we're in a very interesting position for researchers, maybe very, very challenging position for TB control managers and for clinicians. But I think it's something that would create very powerful synergy and would deliver very exciting research results. And I would like to acknowledge New South Wales Mycobacterium Reference Laboratory colleagues that I work with. Elena Martinez is here, and this is Peter and Andrea and Colin, and my team at New South Sydney Public Health Pathogen Genomics Unit, this enthusiastic researchers that made whole genome sequencing of Mycobacterium tuberculosis in New South Wales possible. And thank you very much for great support. Thank you.
Great.
Thanks, Vitali, for a wonderful presentation. Do you have any comments about the interface between the laboratory and clinical management of patients? So at the moment, I think you've done a great job in synthesizing the information for the people in New South Wales to look at, but how do you think labs can better communicate the complexities of this issue when clinicians make decisions about drug management?
Thanks, Greg, for this question. From a lab perspective, our major challenge is to get this testing accredited, because strictly speaking, we can't issue conventional reports to clinicians without accreditation. And hopefully we'll be able to do it in the near future. We issue reports for MDR-TB cases for very selective audience, for expert clinicians that understand this kind of much better than other colleagues potentially. There will be a big challenge for us to educate, to raise awareness, and to educate scientists and clinicians in this. But it's something that is happening internationally. I think Australia it's happening in the UK, it's happening in Canada now, at least in some provinces of Canada. So I think it's an interesting challenge and probably separate research project for social science.